oxygen cycle, which we have done in our biology classes. So saprophytes are taking food from dead material or dead organic material. Parasites, they are harmful fungi, and these are the one which are causing diseases in the humans. Although the number of the fungi, they are much less than the other organisms like bacteria and viruses, but they do cause infections. Same way, fungal infections may be seen in other animals and mostly in the plants also. Like you may have seen fungi growing on big tree trunks, and sometimes they are infectious or they are parasitic to that plant. But like mosses on the forest floor, that is one of the examples of saprophytes. Saprophytes are symbionts. Symbionts or mutualists, they are fungi which have a beneficial relationship with other living organisms. And for example, this is what I was talking about a minute ago. That mycorrhizae and lichens, these are some of the fungi which you can see on the forest floor or on the big tree trunks. They have a beneficial relationship with other living organisms, and sorry, the net was not working properly. Can you hear me?
Yes, sir. We can hear you
Oh,
again.
okay. So, sorry for the interruption. Fungi which are harmful to humans, but there are certain fungi which are useful to humans also. So, human can get diseases directly or from their toxins, and even the plants and other animals can get diseases. So fungi, they can rot the fruits and the vegetables and the food material, and they can even destroy certain non-living materials like plastics and wood also. So there are certain fungi which are useful to humans, but there are certain fungi which are Sorry, there is something wrong with the net. So there are steroids and hormones that may be produced in the fungi or from the fungi. Cheese industry is using fungi for cheese production. So fungi, they are used in the industrial um, areas for humans, but uh, regarding the disease area, we have certain fungi which are producing infection on the skin. So dermatology, as I told you in the initial slides, that whenever you are going to a dermatology ward, you may see fungal infections there. So the most important area where you are going to see fungal infections, they are that is skin, because most of the fungal infections, they are superficial fungal infections or subcutaneous fungal infections which are produced in the skin. This is just to show you that this is a sexual reproduction in case of fungus. Um, as we have read this in our biology classes, that rhizopus on bread or mold on the bread, that may produce through sexual reproduction, and you have a positive and a negative hyphae, which is going to mate and form a zygote, and then another hyphae is developed. Asexual reproduction is the most common reproduction, and we call that reproduction as spore formation. So we have spores in case of fungi, and we have spores in case of bacteria, but both of them are different. In case of fungi, this is a product of reproduction, but in case of bacteria, when we are using the term spores in case of bacteria, that is the that bacteria is forming a form or a type of itself in cases of an unfavorable conditions. So bacteria have a vegetative form and a spore form, but in case of a fungus, when we are talking about spores, that is a living form of the fungus, that is the reproduction which forms spores. I'm going to tell you four types of spores in fungi. So just see this picture. This is called a blastospore. A blastospore is that you have a round or an oval fungus, and there is a budding uh, of an other fungal body which is called a spore. So there are no hyphae in this case. So this type of spore, which is formed from a rounded or an oval fungus which is not having hyphae, is called a blastospore. So a good example of this is candida, or the common yeast which we are using in baking. Another one is an arthrospore. Arthrospore is that you have a hyphae of a fungus. Hyphae is a living part of the fungus. So whenever a reproduction is needed, that hyphae forms a number of small bodies which are called spores throughout its length, and each spore is going to form a new hyphae. This type of spore is called an arthrospore. So this can be seen in a hyphal fungus. So the previous one was a blastospore, which is seen in a yeast or a rounded or an oval fungi, and an arthrospore is seen in a hyphal fungi. Arthrospore is that there is a hyphae. The hyphae turns into a number of small bodies which are called arthrospore, and then they are going to detach from each other, and they are going to form a single hyphae again, and this is how they are going to reproduce. This is an asexual reproduction. Another type of spore is chlamydiospore. Chlamydiospore is that there is a hyphae of a fungus, and instead of that hyphae turning into individual bodies or spores like arthrospore, there is a production of bodies or spores perpendicular to that hyphae, and then that each spore can produce a new hyphae. This type of spore is called a chlamydiospore. So you can see the picture, and a very good example is a rhizopus. It is a hyphal fungus, and this is how it can produce spores perpendicular to the length of the hyphae. Then there is the fourth type of spore, which is called conidiospores or conidia. The difference between chlamydiospore and a conidiospore is that chlamydiospore is produced perpendicular to the length of the hyphae, while the conidiospore or conidia or conidiospores actually, because there is no single spore but there are multiple bodies. So they are produced on the tip of the hyphae. Chlamydiospore perpendicular to the length, and conidiospore at the tip of the hyphae. So there are a number of small bodies called conidiospore or conidia on the tip of the fungus. So they are called conidiospores. Same is what we call a fruiting body in some types of fungi. So, if we revise the spores, blastospores, there are certain fungi which don't have hyphae. They only have rounded or oval bodies. Arthrospores, they are formed in a hyphal fungi. Chlamydiospores, perpendicular to the length of the hyphae. And then conidiospores or sporangium or conidia. There are microconidia, small in size, and then there are macro conidia, but both are on the tip of the fungi. Tip of the hyphae. So penicillium is one of the examples of conidiospores. See this, a hyphae having a number of small spores or conidia on the tip, and this is just like the appearance of a flower. Aspergillus is again a type of a fungus having a conidia, but see this, in case of penicillium you have spores which are arranged in a lengthwise direction, and it is like a budding flower. But in case of Aspergillus, you have conidia on the tip of the hyphae, but they are arranged in a rounded form. If you call the appearance, this appearance is like a fan, like a hand fan or a Japanese or a Chinese fan. So to differentiate between these two fungi, because both of them are spore forming fungi, both of them are hyphal fungi. Both of them produce conidia, but in case of penicillium, you have conidia in this form, and in case of aspergillus, you have conidia in the form of a fan. Why it is important to differentiate whether it is penicillium or aspergillus? Because you have penicillium in the environment, and if you see penicillium in a specimen or a slide, it is non-diagnostic because it is not a disease causing fungus. But in case of aspergillus, if you see, again it is an environmental fungus, you have aspergillus in the environment. But if you see aspergillus in a specimen, and you have this kind of appearance, it is a disease causing fungus. So it is important that what you are seeing in the slide, whether it is a penicillium or it is an aspergillus. Aspergillus causes infection in immunocompromised or immunosuppressed patients. So aspergillus is important. It is a deadly fungal infection if not treated properly, and it is produced in immunocompromised patient. So it is important in case of the specimen. It is important that whenever a specimen is taken, whenever the slide is prepared, or whenever the culture is put for the fungi, you have to avoid the contamination from the environment. So if uh, you have any contamination, because you cannot avoid much, because fungus is present everywhere in the environment also. If you see penicillium, you are not reporting that penicillium because it is not a disease causing. But if you have aspergillus, you have to report it. But again, a point to note here is that uh, what is the specimen, from where the specimen you are taking, and what is the condition of the patient. I'm emphasizing this because if you have aspergillus on the skin, it is non-diagnostic, because aspergillus cannot produce infection on the skin. This is that once I got a specimen from a wound of a patient, and on the slide it was full of aspergillus, but you don't have to actually uh, report that aspergillus, because that aspergillus was actually from the environment. So whenever you are taking the sample from that wound or the ulcer, you might have got aspergillus on that swab, or whatever the instrument you are taking the specimen with. So you don't have to report that, because aspergillus cause infection internally. Aspergillus cause internal infections or systemic infections. Aspergillus cause infection in the lungs. So whenever you have a sputum for cytology or histopathology or a microscopy, if you see aspergillus, that may be diagnostic. But just on the skin, if you have aspergillus, you have to ignore that, because it is not a dermatological infection. So these are were some of the practical points regarding penicillium and aspergillus. Mucor is again a conidial fungus. There is a hyphae, there is a small body on the tip of the hyphae. But in this case, there are a number of small spores, and uh, all those spores, they are not just attached to each other, but they are confined in a very thin membrane. If the membrane is ruptured, all those spores can be individually scattered in the environment. This is a special type of spores. So you have blastospores, arthrospores, chlamydiospores, conidiospores, and this sporangium. So this is what we have done till now about the spores. Then regarding the hyphae, you may have hyphae in some of the fungi. Hyphae may be septate or aseptate. Aseptate means that they are hair-like or thread-like, but they don't have partitions. But in case of septate hyphae, you have partitions. Pseudohyphae is a term that means that the fungus is not thread-like or hair-like, and neither it is a rounded or an oval body. It is in between that shape. So pseudo hyphae is that it looks like a hyphae, but it is not actually a hyphae, and you cannot say it that it is a budding fungus even. So septate, aseptate, and pseudo hyphae. I'm going to tell you the example of pseudo hyphae in the later slides. In cases of fungal infection, mycosis is a term used for fungal infection. So whenever you call mycosis, it means that you are dealing with a fungal infection. So conditions which are important for fungal infections are hot environment with increased humidity in the air. Inadequate water supply may be one reason. Poor living conditions, Then super added or concomitant uh, conditions may be malnutrition, certain other infections, immunosuppressions, and then lack of diagnostic and care facilities. This last point is very important in that because sometimes we are just focusing on bacterial or viral or parasitic infections, and we miss the fungal infections. And fungal infections are last in the diagnostic or differential diagnosis, but they are important, and you should put fungal infection last in your differential. If you are getting a diagnosis for bacteria or a virus or any parasites, then it is okay. But if the symptoms are not diagnostic of a fungal infection, and you have ruled out bacterial and viral infections, then you go for the fungal test. So fungal infections are important and they exist. Mycosis may be opportunistic, that is in case of immunocompromised patients. Example, a very good example is Candida or Candidiasis. Then mycotoxicosis means that fungal infections may cause septicemia. So when we were studying septicemia or sepsis in hemodynamics, I told you that sepsis or septic shock is a big term, which means that shock because of an infection. And in that, number one remains bacteria, then viruses, but certain parasitic or rarely parasitic and occasionally fungus can produce septic shock also. And a very important example is aspergillus. There are two fungi which are angio-invasive. Angio-invasive means that they can penetrate the blood stream, and those two fungi can produce septic shock. One is aspergillus and other is mucor. So these two fungi can cause sepsis. If you have candida or you have the other skin infection causing fungi, you may not have septic shock. Only the organism penetrating the bloodstream or circulation can produce septic shock. So these two fungi, aspergillus producing lung infection and mucor producing sinonasal infection, can cause septic shock. Then you may have fungal allergies, that is type 1 hypersensitivity reaction, or you may have type 3 hypersensitivity reaction in the form of skin involvement. Then you may have pneumonitis, that is the inflammation of the interstitium of the lung. And in all these allergic cases, you are going to see one important type of cell, and that is eosinophils. So if there are a number of eosinophils in blood and you have number of eosinophils on slide in case of tissue biopsy, then it is an allergic reaction. In cases of mycosis, mycosis is uh, fungal infection. Fungal infections, they are divided into three types: superficial mycosis, subcutaneous mycosis, and systemic mycosis. Superficial mycosis means that the fungus is only lying on the epidermis. It is not even penetrating the epidermis. Epidermis, I'm not talking about skin as a whole. Superficial means that it is only lying on the skin but still producing an inflammatory response underneath the epidermis. Subcutaneous means that it is penetrating through the epidermis and producing infection directly in the dermis. And systemic means that the spores are inhaled through the nasal cavity. That means that they are coming from the air, they are airborne, and they are going to produce infection in the lungs. So most of the systemic mycosis are lung or respiratory tract infections. We are going to deal with the individual fungi later on. In general, how can the fungal infections they can be diagnosed? So in case of fungus are or fungi, they are a bit bigger organisms than bacteria and viruses. uh, Same like parasites, like you can see parasites on the slide. You don't go for culture for parasites, but in case of fungus, yes, you can see fungi very beautifully on the slide. But if you want to confirm the type of the fungus, you can go for cultures. But uh, serology in case of fungal infections is not developed, because they are diagnosed on the slide, in the fluids, in the tissues. So serology, that is the branch of immunology in which you are dealing with antigens or antibodies. So this is one branch which is not developed in uh, the fungal infections, and less developed in parasites. And in case of bacteriology and virology, it is a much developed branch. But if you keep virology on the first place, that is one part of microbiology where serology is mostly used. So how are you going to diagnose fungal infection? The best way is the direct wet preparation. That means that you are going to take the fluid or the exudate from the infected part, whether it is a skin, or if it is a systemic infection, you may take the sputum or the saliva, and then you can see the fungus directly on the slide in that fluid. Direct wet preparation means that you are not staining the specimen, you are just putting that exudate or the fluid, a drop of that, on the slide, putting a cover slip, and then directly seeing that under the microscope. Because fungus, they are large enough that they can be seen on a low power, that is 10 or 20. And because they have a distinctive shape, you can diagnose the individual fungus also. KOH preparation is that if you have a tissue or a specimen which can produce any hindrance in viewing the fungus, you can use KOH, that is potassium hydroxide. For example, if you are dealing with a fungal infection in nails, or you are dealing with a subcutaneous fungal infection and you take a skin biopsy, because there are other cells also in that specimen, you are going to put that specimen in a KOH solution. A KOH solution is going to kill or dissolve all the human cells, leaving behind the fungus only. So if you take a drop of that KOH solution after putting the specimen, and depending upon the specimen you received, you are going to put that specimen in the KOH for a period of time. For example, if you are dealing with hair or skin, you're going to put that specimen for an hour or so, or for 3 to 4 hours. But in case of a nail, if you get nail cuttings for fungal infection, you are going to put that for 12 hours in KOH. The nail is going to get dissolved, the hair is going to get dissolved, the skin is going to get dissolved, and then you are going to take a drop of the KOH solution, put it on the slide, put a cover slip, and then view under the microscope. That is KOH preparation. The only thing that is going to be left in the specimen is fungus if it is present. So a direct wet preparation and a KOH preparation. Both these are unstained slides. In case of stained slides or stained preparation, number one is hematoxylin and eosine stain. And on hematoxylin and eosine staining, the fungus appears pink or eosinophilic. Any type of fungus is going to show you pink reaction or staining. If you go for special stains for fungus, one is PAS stain, that is periodic, th- that is periodic acid shift stain. So periodic acid shift stain is again going to stain the fungus pink. uh, and if you go further in special stains for fungus, another good stain for fungus is a silver stain, that is called GMS, Gamori methenamine silver staining. But if you call it silver staining, it is good enough. So silver staining, in case of fungus, fungus is going to stain black with a green background. So the entire tissue is going to stain green, and you can see a fungus, prominently black fungus on the slide. So that is diagnostic of that it is a fungus. So in case of stains, hematoxylin and eosine staining. Then in case of special stains, PS is one, and silver staining is the other good stain to stain the fungus. So in cases of tissues, you can go for special stains for fungi. Fungal culture is done on an agar plate, and you are going to get the growth in 7 days. You are not going to put the fungal culture plate in the incubator. You are just going to put the specimen on the fungal plate. You are going to seal that plate so that the humidity remains inside. You are going to put that in a normal temperature in the lab, and you're going to read the culture plate after a week. So most of the time the fungal diagnosis is done on direct wet preparation, KOH preparation, and on special stains. So there is that you are not going further for fungal cultures or serology in routine, because fungus are diagnosed with the name, with the initial parameters. Saborad's agar is the agar or an artificial media or artificial culture media which we are using for any of the fungal um, types. So in routine in our lab also we use Saborad's dextrose agar. So if you are asked for that which type of agar is used for fungal infection, for any fungal infection, you are going to say that it is Saborad's. You don't have to go into the details of the agar, just the name is enough. So this is a Saborad's agar. This is the petri dish having a Saborad's agar. It is an off-white agar. And in case of fungal culture, you are going to put the specimen in the center of the plate instead of putting three types of streaks like the bacterial culture. In bacterial culture, you put a primary streak, then a secondary streak, and then a tertiary streak to pinpoint the bacteria. But in case of a fungus, you put the specimen in the center, you just seal the plate with masking tape on the side, and put that on the room temperature in the lab. So you are going to read this after 7 days or a week, and you are going to see this type of growth. You can do a microscopy from this culture to pinpoint the or name the fungus. And each fungus is having a different type of appearance on the Saborad's agar. See this? Again, this is Saborad's agar with a fungal culture. Saborad's agar can also be put in test tubes instead of petri dishes. This is just to show you the contamination. As I told you that fungal spores are there around us in the environment everywhere. So whenever we are putting a bacterial culture for bacterial diagnosis, we have to be very careful, and we have to work in uh, an aseptic environment or the aseptic area, so that you don't get fungal contamination. So see this. This is a plate, and this plate is having a fungal contamination. So actually, this fungal contamination is actually obscuring the background bacterial growth. So you cannot actually identify the bacteria. So this is not a real fungal growth from the specimen, but a contamination. So this is if we see this type of plate, so that means that this is a contamination. This is the actual fungal growth from the specimen. So if you divide fungi after all this discussion, there are yeast, there are molds, and then there are diamorphic. Yeasts are fungi which are budding fungi. They are rounded or oval in shape, and they reproduced by budding. Molds are which are having hyphae, and they may reproduce by any of the three types of spore formation which we discussed earlier. And diamorphic are those which can exist in both forms at a time or at different times. So you have yeast, you have yeast or budding, you have molds or hyphae, or you have diamorphic fungi. Examples of diamorphic is the best example is Candida. So Candida can exist in both forms, but most of the time Candida exists in a budding form, but may exist in a hyphal form also. You have yeast, mold, and diamorphic fungi. So this is yeast, and this is not a disease causing fungus. This is the yeast is the name we are using for the routine baking fungus, but yeast is also the name of the entire group that
Sir,
is
sorry to
rounded.
interrupt you.
Mhm.
Sir, just 1 minute left.
Okay, so this is yeast. So this is a budding fungi. I'm going to start or continue the lecture next time with this picture